and welcome to today's Basic Science Keynote Plenary Speaker for the CAHR virtual conference. My name is Mark Brockman. I'm an Associate Professor at Simon Fraser University. On behalf of myself and my co-chair, Lyle McKinnon from the University of Manitoba, we'd like to welcome our speaker today, Dr. Lynn Morris. Dr. Morris is a principal medical scientist at the National Institute for Communicable Diseases, NICID, in Johannesburg, South Africa. She's also a research professor and director of the Antibody-Immunity Research Unit at the University of the Witwatersrand in South Africa. Over the past 20 years, Lynn has made significant contributions to understanding the antibody response to HIV infection and has co-discovered one of the most widely used broadly neutralizing antibodies, CAP256 BRC 26.25 monoclonal antibody that's currently undergoing clinical testing for HIV prevention. Her laboratory performs neutralizing antibody assays for HIV vaccine trials and has also played a very pivotal role in proof of concept antibody-mediated prevention trial, or AMP, which we'll hear more about today. So it's my pleasure, again, on behalf of my co-chair, Lyle McKinnon, and I, to welcome Lynn and to hear about her presentation today entitled Antibodies and Prevention of HIV and SARS-CoV-2 Infections.
So I'd really like to thank the organizers for inviting me to give this talk on antibodies and how they can be used to prevent infection caused by HIV and SARS-CoV-2. My name is Lynn Morris and I am based in Johannesburg, South Africa. A brief outline of the talk, I'll mention the epidemics caused by these two viruses and then focus on COVID-19 vaccines and neutralizing antibodies. And then to talk about antibodies and their use as passive immunization, and here focusing on HIV in the AMP trial. And then I'd like to discuss an antibody that we have been involved in developing for clinical testing, and then to talk about HIV vaccines and their development, and then the lessons that we can learn from COVID-19 and to apply to HIV. So this is a snapshot of the global epidemics. On the top is the HIV, and it's been with us for 40 years. So this is in years. And as you can see that there's been a total of 80 million infections and 35 million deaths. And you can see the peak was around, the peak of death was around 2003, 2005, where, over 1.5 million people were dying every year of HIV. And that has been steadily coming down because of the introduction of antiretroviral therapy. But, many of us believe that we're never going to get this truly down until we have a vaccine. The bottom graph shows the COVID epidemic, and this is in weeks because, as we all know, the virus has not been around that long, but has caused a devastating 148 million infections and 3.2 million deaths. And really, if we don't get on top of this quickly and we allow it to continue the way that we've allowed HIV to continue, that's really going to, those numbers are going to continue to increase. And so it really speaks to the importance of getting the COVID vaccines deployed as quickly as possible. But you can see this curve also, waxes and wanes as the virus circulates and moves into different populations, but overall, it is increasing, and so we do need to implement the vaccine program. But the other thing I want to mention is the colors. And as you can see, the red, which is Africa, is really, it's a small blip on the COVID, although, that may be an underestimation. But really what I want to point out is the HIV is largely an issue that's overwhelming Africa. So just to compare these two viruses, they're both zoonotic infections originating in animals. They spread by different routes. HIV causes a chronic infection. The body cannot clear the virus, whereas SARS-CoV-2 is an acute infection. It's generally over in a few weeks to months, although in some people we do know that there are long-term consequences. They infect different cells, and that's because they use different receptors. CD4 receptor in case of HIV and the ACE2 receptor for SARS-CoV-2. And as we know, for HIV, we have very effective licensed antiretrovirals, but no vaccine. And for SARS-CoV-2, we have repurposed and novel drugs, particularly antibodies. But, we have highly effective vaccines for SARS-CoV. So why has it been so much easier to make a vaccine against SARS-CoV-2 than HIV? Well, that's because our body can clear SARS-CoV-2, so our immune response is very effective. So there's a template to work on, whereas with HIV, the body cannot clear the virus because it integrates into the host DNA. And up until, I guess, October last year, we thought SARS-CoV-2 was pretty stable. It is relatively compared to HIV, but we do know, like all viruses, it does mutate. But nevertheless, the fact that it is more stable makes it less of a moving target. The other thing about SARS-CoV-2 is that everybody develops good neutralizing antibodies within weeks of infection, whereas in HIV, broadly neutralizing antibodies only develop after years and only in a minority of people. Another thing about the antibody response to SARS-CoV-2, to the spike protein, which is the target of the antibodies, is that the response is very focused. Whereas in HIV, the response is much more polyclonal. And in fact, the envelope glycoprotein, which is the equivalent of the spike protein, is also a much more complex trimeric glycoprotein. So this shows shows the structure of the SARS spike protein, which of course surrounds the virus and gives it its name, corona. And the important thing here is the receptor binding domain, which is the main target of the neutralizing antibodies. So it's really been quite remarkable to watch the development of COVID-19 vaccines. I've spent 20 years trying to make a vaccine to HIV or contribute to make a vaccine to HIV, and to see vaccines made within a year has just been the most remarkable thing to witness. But of course, they didn't start from scratch. There were existing vaccine platforms. In fact, many of them developed for HIV, and these were rapidly repurposed for COVID-19. The first phase 1 trial started just 3 months after the virus was discovered. That was the Moderna vaccine. And now there are hundreds of candidates in preclinical development. 27 have already undergone efficacy testing, and 8 are approved for use. So this is in contrast to HIV, where after 20 years we have done 10 efficacy trials, but we have no approved vaccines. So really there's a big difference between how we've tackled these two problems. So this just shows the different vaccine platforms that are being used for COVID-19. I think everybody's quite familiar with all of these companies now. So the mRNA vaccines made by Moderna and Pfizer basically take the mRNA of the spike protein and encapsulate it in a nanoparticle, and then that makes a de novo protein to stimulate neutralizing antibodies. The viral vector vaccines basically deliver also the gene for the spike protein into the body, and most of these are using the adenovirus vectors. So this is AstraZeneca, J&J, and the Sputnik vaccines. The viral protein vaccine, so this is a more traditional way of making vaccines. So this is a recombinant protein that's used to stimulate an immune response, obviously much more complex to manufacture, but Novavax has taken this one to efficacy trials. And then there are the inactivated SARS-CoV-2 vaccines. So these are the real old-style traditional vaccines where the whole virus is grown up in large quantities and then inactivated. And so people are actually immunized with all the proteins, as opposed to the other vaccines where they're only getting the spike protein. So it's clear that neutralizing antibodies are important for protection. And in fact, all the vaccines elicit neutralizing antibodies to the spike protein. And some of these are at higher levels than infected individuals. There's evidence from animal studies that have confirmed that neutralizing antibodies are the primary mechanism of protection. Some recent nice data from the VRC using the Moderna vaccine. But we, of course, also had this from the passive transfer experiments with the monoclonal antibodies. But we are still awaiting a correlates of protection analysis from all the human trials, and that's really important that we get this information because we need to know the level of antibodies that are required to prevent infection, so that we can monitor that to see what people's risk is of becoming infected as the vaccine titers wane, and also about whether people need to be revaccinated, possibly every year or every couple of years. But we need to know what the levels are that are needed for protection before we can make any of those decisions. So this is a very nice slide that was put together by the Bill and Melinda Gates Foundation, and it shows the levels of neutralizing antibodies to all of the vaccines that have undergone efficacy testing, and they ranked according to the levels of antibodies compared to what's seen in convalescent plasma. So the Novavax, the subunit or the protein vaccine, induces the highest levels of neutralizing antibodies. They're fourfold higher than convalescent plasma. It is also the most efficacious vaccine, as you can see there, 95.6%. The next two are the mRNA vaccines, which are, as you can see, 3.4 and 3.8-fold higher than convalescent plasma. And these are also highly effective vaccines. The next group are the vectored vaccines. So the AstraZeneca induces equivalent levels to convalescent plasma, whereas the J&J vaccine actually doesn't induce as high levels as convalescent plasma. But as you can see, they're pretty similar in terms of their efficacy. And then there's the inactivated vaccines, which there's some differences here, but the one that has the lowest levels, so the Sinovac, which has sixfold lower than convalescent plasma, is also the least efficacious of the vaccines. So, of course, what I've been talking about is vaccines that are targeting the original Wuhan strain, but now we know from studies around the world that this virus mutates. And we know that these variants of concern have emerged in different regions of the world. And I'm going to focus here on the one that's emerged in South Africa, the 501YV2. So this was first detected in South Africa in October by Tulio de Oliveira, who does genomic surveillance, and he noticed some mutations that he thought were going to be important, and he started tracking it. And as you can see from this graph, that the yellow bars here, it started off small, and by mid-December, in fact, this variant had completely replaced the original variant. And he was able to, in fact, trace it down to a region in the Eastern Cape province of South Africa, but it quickly spread to other provinces and continues to be the dominant virus in South Africa. So this virus contains a number of different mutations, or a number of mutations in the spike protein, but the ones that are of most importance are in the RBD. And so that's the 417 and the 484 mutations. Of course, the 501 we know increases the transmissibility of the virus. But certainly these two mutations looked important in terms of potential immune escape. And so this was tested in the laboratory, and this was work that has been set up by Penny Moore, who very quickly pivoted our HIV pseudovirus neutralization assay to measure antibodies to SARS-CoV-2. And so this shows the levels of antibody. This is a cohort of hospitalized patients, of patients hospitalized with COVID, and their titers to the original strain. So I should say these are samples that were collected prior to the emergence of the variant. So they were infected with the original variant. And as you can see, they have, most have pretty nice titers. But as you can see, they were less effective at neutralizing this variant, this 501YV2. In fact, there was a 9-fold reduction in the ability of these antibodies to recognize this variant. And in fact, 48% of them, there was a complete knockout. So this raised immediate concerns, or for the potential for reinfection, but also for vaccine efficacy. And indeed, that proved to be the case. So this is some specimens from Shabir Mahdi's small clinical trial of the CAHR vaccine. And you can see that people who received this vaccine were capable of neutralizing the original variant, but they were really very ineffective at neutralizing this 501YV2 variant. And unfortunately, this trial was being done at the time when this variant had started circulating. And in fact, when the trial was unblinded, there was in fact no efficacy. So this was a 2-dose regimen of the CAHR, and there was no protection against mild, moderate COVID due to this variant. And they did sequence the breakthroughs, and the majority of them indeed were this variant. So clearly here, the laboratory data was very predictive of the clinical outcome. I should just say, though, that we don't know about protection against severe disease because this trial was not designed to test that. And since this vaccine is actually not being used in South Africa, it's not going to be a question that we'll be able to answer. So another important thing was, well, what is the immune response, or what is the antibody response to this variant? So people who are infected with this variant, what do their antibodies look like? So this is just the same data I've just shown you with the reduction in the ability to recognize this variant. But this is the individuals who are infected now with the variant and their ability to neutralize the original virus. And as you can see, there's a slight decrease. It's only threefold, but importantly, only 7% of them have a complete knockout as compared to almost half of them in the infected with the original variant. So really a big difference there. And what this suggested was that people infected with this variant, in fact, had cross-neutralizing activity. They were able to recognize, the original variant quite effectively. And in fact, this was also shown when Tanteka tested these samples against the P1 variant, or the 501YV3, which was originally circulated in Brazil. And as you can see, there was in fact no loss of activity. In fact, it actually neutralized this variant a little bit better. And so clearly these antibodies are cross-reactive. So just to summarize this part of the talk, neutralizing antibodies correlate with SARS-CoV-2 vaccine efficacy. They correlate. That doesn't mean it's a correlate of protection. Those analyses still need to be done. But the 501YV2 variant shows substantial or complete escape from neutralizing antibodies in COVID-19 convalescent plasma, vaccine sera, including CAHR and actually the J&J, although I haven't shown you that data. Therapeutically relevant monoclonal antibodies, and again, I haven't shown you this, but it is in the Nature Medicine paper if you want to look at it. And that obviously also has implications for the use of these monoclonal antibodies in a setting where this variant is circulating. Importantly, the sera from people infected with this 501YV2 showed good cross-reactivity and neutralized both the original variant and this P1 variant. And this has positive implications for vaccine design. And in fact, many of the developers are already starting to incorporate these other spikes into their existing platforms. And certainly the mRNA vaccines, I think, have a good advantage here in being able to do that very quickly. So now I'd like to talk about passive immunization, so the use of antibodies as biological drugs. And this is really a faster way to deliver immunity. You don't have to wait for immunity to develop, which of course takes up to at least a month after the 2-dose vaccines. So in this case, the infusion of antibodies would be before or shortly after infection, and then would provide instant immunity. So again, the technologies that were developed for HIV, the structural biology, the single B cell sorting, and the amplification of the antibodies was developed that were developed for HIV really paved the way for the rapid isolation of antibodies to SARS-CoV-2. And already, and I'm sure there are many more than this, antibodies in development. There have been 5 efficacy trials and 2 have already received emergency use authorization. So these haven't been widely used, and that's probably because there's efficacious vaccines, but they are likely to be used in specific situations, and particularly for prophylaxis in outbreaks. They've been shown to work very effectively, particularly in people who are highly vulnerable, such as old age homes, where you need to get people protected quickly, but perhaps also in early infection. And in fact, they certainly have been shown to be to be effective in that setting as well. I want to talk about passive immunity in HIV, because we've just completed this AMP study, the antibody-mediated prevention study, which was published in NEJM last month. And this is testing VRC01, which is a broadly neutralizing antibody that binds to the CD4 binding site on the HIV envelope trimer. It's very broad. It neutralizes 90% of global isolates. So this antibody was isolated in 2010 by the Vaccine Research Center. And again, just to once again highlight how remarkable the progress of COVID has been, that in a year they had antibodies isolated, clinical trials done, and being used in a year, whereas it took us 10 years. So we clearly need to learn from that experience. But just to tell you a little bit about the trial. So it's in fact 2 trials. It's a collaboration between the HIV Vaccine Trials Network and the HIV Prevention Trials Network. So the Americas trial was in men who have sex with men, and the Africa's trial was in heterosexual women. So participants either got a low dose of VRC01 or a high dose of VRC01, and there was a control group. In total, there were 4,600 participants, and they received 10 infusions, and these were intravenous infusions, and there was one every 8 weeks. And so the reason for that is because the antibody decays, and so in order to maintain protective levels, fresh infusions were given every 2 months. And so this trial continued for over 4 years. And really was just a remarkable trial to have done. The retention rates were incredibly high, and really just an exceptionally well done trial. So perhaps somewhat disappointingly, there was no overall protection in this trial. And the results are shown here both for the 704 and the 703, but since there was no difference between the trials, I'm just going to focus on the pooled data. So as you can see, there were 67 infections in the placebo arm, there were 60 in the low dose and 47 in the high dose, so overall 107 in the pooled. But you can see these lines overlap 0, so no prevention efficacy, and as you can see the p-values as well. But this trial was always about the sensitivity of the viruses. So what we were doing in the laboratory real time was measuring the sensitivity of the breakthrough viruses. And so samples from infected participants went to Carolyn Williams's lab in South Africa and Jim Mullins' lab in the US, where they identified the transmitted founder viruses, and these were synthesized, and we made pseudoviruses, and then we tested them against VRC01. So obviously we were doing this blinded, but our hypothesis, was that in the VRC01 arm that they would mostly be resistant viruses, and in the placebo arm they'd mostly be sensitive viruses. So that was the hypothesis. So we were able to measure the in vitro sensitivity of 162 of the 174 endpoints, so 93% of the viruses we were able to assess. And we used pre-specified categories. So viruses that were very sensitive to VRC01 at less than 1 microgram per mil were called sensitive, and then between 1 to 3 micrograms were intermediate, and then greater than 3 micrograms were considered to be resistant. And so to just focus on the cyan, because that's really the only area that we saw a difference, You can see that in the placebo arm there were 19, which corresponded to 30% of the viruses, were very sensitive to VRC01. But in the two treatment arms, there were far fewer viruses. So these viruses were essentially blocked by VRC01. So in total, there were 9 viruses in the pooled. So that's shown here in the analysis. The statistical analysis showed that indeed there was prevention efficacy in viruses that were very sensitive. Here's the 19 and the 9, and it's 75% efficacious. There was no efficacy in the intermediate or the resistant, or the viruses that were resistant to infection. As you can see, these lines overlap 0. So this is just another way to look at the data. So this shows each individual virus. So this is the IC80, or the concentration that causes 80% inhibition. So you can see there's a huge range, but the viruses that are super sensitive are the ones below this line, and you can see there's quite a number here in the placebo or the control group, whereas there's far fewer in the VRC01 arms. So indeed, this proved our hypothesis that that the viruses in the treatment arms were more resistant to VRC01 than in the placebo arm. In fact, they were 2.4-fold more resistant. They had greater IC80s because basically these viruses were blocked by VRC01. So in summary, the prevention efficacy against viruses that were sensitive to VRC01 was 75%. However, there was no overall efficacy in this trial, and that's because only 30% of circulating viruses are sensitive to VRC01 at that level. I think another very important thing about this trial is, there was no impact on gender. As you know, the trial included men and women from different countries and dealing with different clades of virus, clade B in the US and clade C in Africa largely. And no difference between the doses, the high or the low dose, or the route of acquisition on prevention efficacy. Another very important outcome of this trial was that the in vitro sensitivity that we measured in this TZMBL neutralization assay was in fact highly predictive of in vivo efficacy, and that's really important because it means that that what we're measuring in the test tube is telling us what's going to happen in clinical trials, and that's important for moving forward. So, AMP did not show overall efficacy, and should we be disappointed? So Bruce Walker wrote a very nice editorial, and he titled it "A Half Glass Full," which I thought was very appropriate, because there has been some people, have been, very excited about the results, and some less excited. I'm certainly in the half full category. I'm an optimist. But also, it means that we can fill the rest of it. We can reach the full potential because now we know where we're headed. And we need to remember that VRC01 was the first available BNAB. It was never intended as a product. But this trial has provided us with the signal we need to move forward. And indeed, we are moving forward with other BNABs. And in fact, the newer BNABs are far more potent and broad than VRC01. And in fact, we also now know from in vitro experiments that these combinations can increase coverage. So this is just to give you an idea of some of the other targets. So we've been talking about VRC01, which is the CD4 binding site. So this is recessed on the envelope trimer. The other CD4 binding site antibodies that are being considered are listed here, and I'll be certainly talking about VRC07523LS a bit more. But there are 3 other targets. So the V2 apex, and these are 2 antibodies that are in clinical development. I'll talk about CAP256, and then the V3 glycan or the N332 glycan supersite targeting the V3, 2 antibodies there, and then an antibody in the MPER. So this just shows all the antibodies that are in clinical development and what stage they're at. So these just represent different versions. Some of them have been manufactured to increase some of their, to increase some of them have been engineered to increase their manufacturability. And of course, some also have the LS mutation added on to increase their half-life. But this just shows the various combinations that are being tested. And you can see these are phase 1, many are ongoing, some have been completed. And by a number of different organizations and a number of different combinations. It's dual or triple combinations, of antibodies, using also both the IV and the subcutaneous routes. Also, just to mention, there are two a bispecific antibody that is currently in phase 1 clinical trial that combines the properties of an IMAB, which targets CD4, and 10E8E4, which is a very, very potent bispecific antibody, and there's also a trispecific antibody as well. So I want to talk now just a little bit about CAHR2021, because this is an antibody that we were involved in, we identified the participant in the CAPRISA trial who had very potent plasma antibodies, and we teamed up with the VRC and isolated this very potent antibody called CAHR2021 VRC26. And as you can see here, the median IC50 is indeed very impressive. It is one of the most potent antibodies. It's not the broadest because it also is very specific for clade C. One of the other unusual features about this antibody is it has a very long CDRH3, and that really is in fact how it manages to be so potent. So this antibody has been manufactured for clinical trial, and I should also say we've also shown together with Dan Baruch that it protects monkeys at very low dose. So that the high potency translated to a low dose in vivo dose and protective, as I said, at very low dose. And it's been manufactured, and the phase 1 and the PK trial is underway. So just to tell you that, taking these things through this GMP process is not easy. And in fact, this particular antibody, because of its long CDRH3, turns out it was sensitive to proteases in the cell line. And Nicole Doria-Rose did a fabulous job of figuring out that the target is a lysine in this CDRH3, and so she changed it to an alanine. Unfortunately, it had no impact on the neutralizing activity, but this did cause an 18-month delay in the manufacturing process. But clearly, obviously was well worth the persevering to sort out. So in terms of the trial, so the CAPRISA-012b trial, this is a first-in-human trial to assess the safety and PK of this antibody, which is called CAHR2021 V2, version 2, and LS because it has the long half-life mutations in the FC domain. The first injection was on the 13th of July, 2020, last year. It's being tested alone and in combination with VRC07523LS, the CD4 binding site antibody. It's a dose escalation study using intravenous and subcutaneous routes of administration. And the subcutaneous administration is made possible through the use of hyaluronidase, which depolymerizes hyaluronic acid, and that increases the dispersion and absorption of the antibodies. And it also allows much larger volumes to be injected. So this is going to be a game changer for using antibodies on a bigger scale. So in terms of an update on where we are with the trial, there's been 2 DSMB reviews, and the antibodies been shown to be safe and tolerable. There have been no major safety concerns. The PK levels are very similar to other BNAbs, and we were obviously delighted to see that, given that this is an unusual antibody. And then the predictive modeling suggests that at 20 mg per kg at sub-Q administration, that the serum concentrations of 1 microgram per mil will last up to at least 16 weeks. And so what that means is that injections could be given every 4 months and perhaps even every 6 months, because we obviously don't know. Well, we need to find out what the PK is long term, but certainly that's the hope, that an injection perhaps every 4 to 6 months could protect, and this is being designed specifically for young women who are at high risk of contracting HIV. And so the other thing, so as I mentioned, this antibody is being tested in combination with VRC07, and so this is just some theoretical, the theoretical coverage using the cutoffs from the AMP study. So at an IC80 greater than of less than 1 microgram per mil, we would predict that this combination would neutralize 89% of a multi-clade virus panel, but in fact 97% of clades of 58 viruses in that panel that are clade C, because we do know it's better on clade C, and then also on a acute clade C panel 90%. So pretty good coverage for this dual combination. So what does the future hold? Well, AMP has provided this proof of concept that bNAbs can be used for prevention. As I've mentioned, we have better antibodies and bispecifics. We have this in vitro biomarker that's going to be very useful for deciding which antibodies to move forward with. Definitely the manufacturing is getting cheaper and more efficient, and that bNAb combinations show high coverage of clade C. And this provides a strong, so I should also say this, the AMP study provides a very strong rationale for pursuing vaccines that induce broadly neutralizing antibodies. So now I'd like to turn our attention to talking about HIV and vaccines, and why there's still a really urgent need to develop an HIV vaccine. So in 2019, we still had 38 million people living with HIV, 700,000 HIV deaths every year. And this is the big one, 1.7 million new infections. We're just really not impacting enough on incident infections. And of course, as I mentioned, a large of these are, in Africa. And sorry, just to say as well, we're talking about almost 5,000 infections every day. So we have done 10 efficacy trials over the last 20 years. I'm not going to go through all of them. I'm going to just focus on the ones with the green ticks, which there are 2. So the AMP study we've already discussed. That's over here. But a trial that was completed over 10 years ago now, the RV144, which used a canary pox vectored vaccine as well as a protein GP120 boost. It was done in Thailand in over 16,000 people. Showed a 31% efficacy level, and this vaccine was in fact an analogous vaccine was then tested in an efficacy trial in South Africa, and I'll show you those results. In fact, this is the trial over here. So the only one also that we're still waiting the outcome of is this HVTN 705 trial. But as you can see, there are a lot of crosses here, and this is obviously very different to the coronavirus landscape, where almost every vaccine that's been taken to efficacy testing has worked. So this really speaks to how difficult an HIV vaccine really is. But I also think that we should be populating this more. We should have many more trials on here, and we should be trying a lot more vaccines and doing them all at the same time. So this just shows you the results from the RV144 trial, the trial that was done in Thailand, that showed overall a 31% efficacy at 3 years. At the earlier stages, when the numbers were very low, there actually was a suggestion that it was even more, efficacious early on. But the important thing from this trial, and surprisingly, it surprised a lot of people, is that when the correlates analysis was done, the correlate was a non-neutralizing V2 antibody, everyone had assumed that we would be stimulating neutralizing antibodies. But nevertheless, this led to what's called the 702 trial, which is really building on the or trying to replicate the RV144 trial, but in Africa. And so this trial started in October 2016, and it used, again, a canary pox, but it was modified to include clade C inserts, and also the subtype C, a subtype C GP120 with a different adjuvant. But this was done in over 5,000 South African men and women at high risk of HIV infection. So the aim of this trial was to test whether non-neutralizing antibodies are potent enough to achieve vaccine efficacy of greater than 50% for at least 2 years. And very disappointingly, this trial actually was stopped early because of futility. As you can see here, the vaccine and the placebo arm overlap. And this was just, again, published in NEJM last month. And so clearly we need to understand why this trial didn't work. It's very similar to the vaccine that was tested in Thailand that did show an efficacy signal, but of course there were lots of things that were different. The population is different, the routes of transmission are different, and of course there were differences. Even though the regimen was similar, there were some differences in the inserts and certainly the adjuvant as well. So the trial that is still going on, one efficacy trial is the HV10-705-706, which is in fact using the same vector. It's by Johnson & Johnson, same vector as the Johnson & Johnson vaccine being tested for, that's shown to be effective in COVID. This obviously contains, an HIV insert, and in fact it was a bioinformatically selected insert and designed to cover all of the major viral subtypes. And then there's a boost of a GP140, a clade C GP140. So this vaccine is designed to elicit non-neutralizing antibodies. The rationale for taking this to efficacy trials is that it did show very good protection in non-human primates, and we are expecting these results late in 2021. But, of course, I think most people do believe that an efficacious vaccine will require the induction of broadly neutralizing antibodies. And this is a very tough challenge. But what has been very helpful is, has guided the field really, is the studies of natural infection of those people who do develop broadly neutralizing antibodies. So roughly 20% of people develop some level of breadth, but the real elite neutralizers is only about 1% of people. But this does take many, many years. And it really is about the co-evolution of the virus and the antibody, how the antibodies change to counter the virus, and then the virus escapes and the antibody then changes. And so it really is this ongoing co-evolutionary process that results in broadly neutralizing antibodies. And indeed, some of this analysis has gone down to individual amino acid level about the kinds of changes that one might need in order to elicit the right kinds of antibodies. But it also starts right here at the beginning with the engagement of what is called the UCA, or the germline antibody, the right B cell that's going to be able to develop the right kind of antibody. And finding these precursors, because they're very rare, is also not easy. And so these are some of the newer approaches that people are trying. So this germline targeting is being championed by Bill Sheaf, and basically to try and fish out the very rare the precursors that are going to give rise to CD4 binding site antibodies that use this particular heavy chain gene, the VH1-2. And so he's developed this outer domain with these nano particles, but basically it engages particular germline B cells. And in fact, just at R4P, he showed that he has been able to do this in humans. So humans, he has immunized humans and been able to show that you can select for the right germlines. Of course, the next step would be to expand them and obviously boost them. but certainly as a first step, that's very encouraging. Another very active area is the use of trimers, so trimeric proteins, to stimulate broadly neutralizing antibodies. A lot of work has been done in animals to show that you can certainly get neutralizing antibodies to the vaccine strains, but getting broadly neutralizing antibodies still remains a challenge. But certainly there are clinical trials now in humans with these trimeric proteins, and we look forward to seeing how those pan out. But also just to say that people are trying, to mimic what happens in natural infection with the evolving virus, so to also, have immunogens that are slightly different in order to shepherd this antibody response along a pathway to breadth. So in conclusion, vaccines for COVID-19 and HIV are urgently needed for public health, economic and societal reasons. And I think we all accept that, it's clear that the vaccine to COVID-19 is going to help us get out of the COVID-19 epidemic, and we're already seeing very positive signs of that. But, COVID-19 vaccine and antibody development benefited from decades of HIV research. And COVID-19 has changed the way we do science. And so the hope is that this, what we've learned will help speed up the development of a much needed HIV vaccine. And, here's just some of the strategies that were used to develop a COVID vaccine really quickly, testing multiple vaccines, doing it concurrently, rapid sharing of data, getting these approvals really quickly, new approaches to funding and research collaboration, early manufacturing, often at risk, and then, of course, just this global sense of urgency that we really do need to revive for HIV. So what are the lessons that we've learned? Well, that vaccine development can be very fast, and that there's no doubt that safety is the most important criteria and cannot be compromised. I mean, we've seen what happens when there's, safety signals. Everything grinds to a halt. So the safety is the most important thing. Cooperation with industry is key. We've seen a lot of companies come on board in COVID, and we hope that we could engage them as well in the HIV vaccine development. Definitely strong leadership and globalism. These are global problems, and of course the funding is a critical aspect for attracting all the brains to apply to this problem, and also about how we need to counter misinformation and vaccine hesitancy. So with that, I'd really like to thank this wonderful team of people led by Penny Moore, sitting over here, and who's just, really taken on board the COVID challenge, and, people have been working extremely hard, and just really to thank all of them, Tandeka, Kurt, Janelle and Mashudu, but also to thank Nono and Tandile and others who've worked on the AMP trial, which really was a very important trial as well for the lab, but really to thank the whole lab for the great work. And then to thank all of our collaborators, particularly Slim, who's really led the testing of the Caprisa antibody and shown great leadership in taking that forward. Longtime collaborator Carolyn Williamson and David Montefiore. Of course, the HVTN for having the vision to do the AMP study, Larry and Mike. All of the statisticians and the great work that they do. We don't always understand each other's languages, but we get there in the end. And of course, Jim for the sequencing work. And then the very rewarding and long-time collaboration we've had with the VRC and some of our other colleagues at NICD. And then, of course, our clinical collaborators on the COVID work in terms of convalescent samples and also vaccine samples from Shabir Mahdi. And then really to thank all of the AMP study trial participants. What an incredible trial, and the real commitment that they showed to seeing this trial to the end. And with that, I thank you all very much.